All right. Thank you for joining us today. And also thank you for your interest in Nanorobot for biomarker instrumentation of cancer. And in today's presentation, we are going to cover our study, right? Which basically what we are doing is using computational tools, right? Or name it computational nanotechnology with clinical data towards prototyping of nano devices for cancer, right? Nanomedicine instrumentation. Of course, nanotechnology is a challenging topic, right? Particularly nanorobots. And let's say patience and determination is a key, right, when it comes to developing nanorobots. And we are going to cover, right, so things such as computer-aided design, right, simulation, as a tool for prototyping, right? We will discuss or briefly mention about trends, and then we jump into case studies, right? And from there, we are able to draw some conclusions. One thing is that is quite often mentioned is computational nanotechnology, right? And as a matter of fact, computational nanotechnology is a very important tool, right, as a scientific method and also as an approach for implementation and modeling that actually bring together applied math, computer science, right, towards application to solve engineering problems, right, and also natural science investigations, right, and particularly in our case, we have interesting biomedical application of nanotechnology, right, and one thing, of course, that is important is when you're doing, let's say, applied research, right, to think in terms of, okay, how we can visualize that this investigation might result in products, right, that will benefit the marketplace, right, either on the short or on the long term, right, and of course bringing let's say experts with different background, right, as an interdisciplinary team is very important. And one way to achieve a practical development of, let's say, prototyping of nano robots or nano devices is to work with case studies, right? And why? Because it helps us, right, in terms of providing the criteria, right, and the parameters that we need to establish when collecting the data, right, for whatever modeling, right, we have in mind. And of course, it's important that we try to achieve, right, effective conclusions, right, from the subject we are investigating. Other thing also to keep in mind is that many people visualize software and hardware, right, as not directly related, but as a matter of fact, software is a very effective tool, right, helping to build hardware, right. In terms of miniaturization, right, everybody is well aware that things are getting smaller and smaller, right, and from that concept, we came to the idea, right, of thinking in making the investigation and prototyping of nanorobots, right, for medicine, right. Of course, we include in the model and in the computational simulation, right, all the biomaterial, right, necessary for teleoperation and integration of nanoelectronics and sensor activation, right, and of course, when it comes for biomedical application, right, is even more important to consider clinical data. In terms of approach for nanorobot development, right, there are, let's say, a variety of approaches, and the one that our team find more favorable is using nanoelectronics, right? And in that aspect, right, we adopt computer simulation, right, as a computer-aided manufacturing approach, right, towards modeling and prototyping of nano devices, right? And of course, we need to consider, let's say, the nanoelectronic circuit model, right, and also all the materials necessary to allow us communication with the device, right? Also tracking and activation, right, of the nanorobot itself. It's also important to keep in mind that the nanorobot needs to be self-contained, meaning that all parts have to fit, right, as an embedded device, right, for the effective application of nanorobots for fighting cancer, right? Other thing also to, of course, keep in mind is the fact that 3D modeling, right, help us as an applied approach, right, and methodology for our research, right? So we use case studies, right, as the most, let's say, feasible way to investigate development of nanorobots, right? And it's obvious that many companies use the same approach, right? So like airplane companies, right? Also automobile, right? And aerospace industry and the medical industry, right? So when we talk about manufacturing, right? Manufacturing and computer-aided design are always working and walking together, right? So, but for cancer specifically, right, the idea is we want the nanorobot to be able to detect if, let's say, a patient has some tumor in early development, and for that we consider the protein MYC as an overexpression, right? That is helpful in terms of, let's say, detect if we have in a certain part of the body, right, any, let's say, tumor in early stage of development, right? So, and of course, the MYC protein, right, has been proven as one of the proteins that are directly related and linked to cancer development, right? And thus we can use basically the chain of gradient, right, within the bloodstream, basically to allow us to activate the sensor, right? And once the nanorobot sensor sense and detect that we have a change in the gradient levels, right, of the protein MYC, then the nanorobot sensor is activated, and the nanorobot sends a signal, right, pointing out or pinpointing that we have overexpression of the protein MYC, right? So it's also noteworthy to mention that we adopt the threshold of 100 nanorobots, providing, let's say, a validation against false positive, right, for the protein MYC in the bloodstream, right? So again, the whole concept is to use the nanorobots, right, as collective swarm sensors that goes foraging and searching for a pathological signal, right, in the patient body, right? And of course, doing the computer-aided simulation, right, for nanorobots is a quite effective way, right? Because basically this provides us very insightful information towards manufacturing analysis, right? And one thing also interesting to note is that the same approach, right, we have for cancer instrumentation with nanorobots using protein, right, or proteomics information, right, such as the MYC protein overexpression. This same approach can be also applied for, let's say, heart problems, right, cardiology problems, in for example, coronary arteriosclerosis, and practically the same concept, right, and design is applicable also for other problems, right? So not only heart and cardiology, but also diabetes, right? We have a related protein that can be used for the glucose monitoring, right, in the bloodstream, right? And even for homeland security, right? Right, for defense, for example, right now we have the pandemic, right, the coronavirus, right? And similarly, we can consider the same idea, right? So we have implemented a study case before for influenza, right? And basically the concept again is that we need to consider what is the material, right? What is the protein overexpression and from the proteomic analysis, right, and the materials, we are able to define what is the antigen for the sensor, right? And thus, if a patient is infected, right, we are able to pinpoint that the person has a let's say, exceedingly high signal, right, for that specific pathological problem, right? Additionally, we also can apply nanorobots for brain aneurysm, right? And again, depending on the application you have in hand, right, will be the protein that you want to include in the model, right? And of course, when it comes to working and implementing, let's say, a model for a nanorobot application in cancer, right, and also in other biomedical problems, and overall, when we talk about nanotechnology, right, of course, determination is very important, right? So, we should apply that, right, for anything, right, but specifically for nanorobotics and biomedical application, right, in terms of nano devices. The other thing also is, in terms of conclusion, three things we need to keep in mind, right, is always think outside the box when it comes to nanorobotics for cancer. Also think in terms of having a multidisciplinary team collaboration, right? And of course, to be very determined, right? Be very determined in the investigation and implementation of such devices and models, right? So basically, these are all the topics we have covered, right, in this presentation, right? So we hope you have enjoyed, right? And of course, if you have any question, right, please feel free to ask. We hope, again, you have enjoyed the presentation, right? And basically, see you next time.